Hello everyone, welcome to today's webinar. I'm Kübra, a global marketing specialist from IVIM Technology, and I'm thrilled to see such an enthusiastic turnout for our session. Today we will delve into the latest advancements in live animal imaging, focusing specifically on real-time intravital microscopy and in vivo cellular level imaging in the fields of immunology and neuroscience. To provide you with a comprehensive understanding of this cutting-edge technology, we have prepared a live demonstration as well. Throughout the webinar, please feel free to ask any questions you may have using the Q&A box below. We will make sure to address as many questions as possible, including those that were submitted prior to the webinar. Without further delay, I'm excited to introduce Dr. Selina Soyeon Ahn, our esteemed presenter. Selina holds a PhD in Intravital Imaging Research from the Korean Advanced Institute of Science and Technology. With over 7 years of experience in the field, she currently leads the Field Applications Specialist team at IVIM Technology. Selina, please take it away and begin with your presentation.
Thanks, Kubra, for the kind introduction. Hi, I'm Selina. I'm an application specialist in IVIM Technology. It's my great pleasure to meet you, even it is virtual, but very happy to introduce our technology to you today. And for today's presentation, I'll briefly introduce our technology and intravital research applications, especially in immunology and neuroscience research fields, which have done by using IVIM system. Please note that the following contents in the presentation contain animal illustration, not the real animals movie utilized in each in vivo research. During the webinar, if you have any question, please text it in the Q&A box. Then I will respond to your question on the Q&A session as soon as possible. Firstly, I'd like to introduce IVIM's intravital microscopy to observe live tissue and cells in live animals. Compared to a conventional macroscale imaging technology such as X-ray, CT, and MRI, which only provides the tissue level and the static in vivo information, either way, the intravital microscopy can directly observe dynamic cell movements inside of the internal organ of the live animal model for the human disease. So this example video shows how red neutrophils inside of the vessel can form a cluster and block the green blood circulation in the lung of sepsis animal model. So intravital microscopy can provide a detailed live cell video of the disease development in the cell level. Especially, we've developed a specialized device for the stabilized lung imaging, which is the suction-based system, which are optimized for in vivo lung imaging. We expose the lung tissue through the rib incision, then with the little negative pressure applied through the suction hole of the lung imaging window chamber. Thereby, we can let the lung tissue to be attached to the cover slip in a stable manner. So with this in vivo stabilized lung imaging system, we can acquire this kind of high resolution microscopic images of the neutrophil dynamic in the pulmonary vasculature in the live mouse model. And not only the cell, but also we can image the drug actually. This is a 24-hour image we obtained from the cancer implanted mouse model. So in this data, the GFP expressing human breast cancers we've implanted to the dorsal skin have just grown to a certain site of a tumor after 2 weeks of implantation. Then we treated the red fluorescent nanoparticle drug to analyze the drug delivery and the distribution to the target breast cancer. Within 24 hours, we can see that amount of the nanoparticle get out from the vessel, then finally fully delivered to the breast cancer tissue. So this is one of the very nice example how we can utilize intravital microscopy imaging technology to monitor the drug delivery to the target cell, target tissue, or the organ in the live animal model. Conventionally, for intravital experiments, users have to prepare each required functions by themselves. There was a high degree of the technical difficulty and non-optimized performance with the limited application to imaging interest organs without any standardized in vivo protocols. So IVIM's intravital microscopy system is extensively optimized and carefully engineered to provide the best performance in the intravital imaging of the live animal model. IVIM's intravital microscopy enables 3D cell-level microscopic imaging and analysis in vivo. It successfully achieves the dynamic intravital cell imaging, such as real-time cell trafficking, cell-to-cell interaction, and cell-to-microenvironment interaction inside of the living body. And furthermore, it can be widely applied to the therapeutic research fields, investigating the drug delivery to the target tissue, drug efficacy, and therapeutic mode of action validation in various preclinical animal models of the human disease. IVIM is the world's first and only one company providing new all-in-one system with the best in vivo imaging performance. So this all-in-one single box package enables easy installation, operation, and maintenance, and it has co-optimized hardware and the software for the fast in vivo imaging performance with ultra-fast imaging speed up to 100 frames per second. Thereby, we can even capture the very fast cell dynamics, such as red blood cells that are flowing inside of the blood vessel. And together with the automatic hassle-free motion compensation function and various accessories such as live animal maintenance system and various imaging window system for the wide range in vivo applications, users easily can get the high quality in vivo data without any limitation in the imaging target organs. So here are the in vivo imaging window chambers that are fully optimized for the target organ-specific applications, especially for long-term monitoring. Dosal skiffle chamber is for in vivo research of the cancer and the drug candidates. And for intravital heart and lung imaging, we utilize the suction-based tissue motion stabilizer window chamber system to temporarily stabilize the imaging side of the beating heart and the lung tissue. And in case of the brain research, we implant the cranial imaging window in the skull to visualize the brain cortex in the long-term manner. And abdominal imaging window is for abdominal internal organs, such as pancreas, spleen, kidney, and colon, etc. For long-term memory tissue and pancreas imaging, we could utilize the memory and the pancreas imaging window chamber each. And for uterus imaging window, uterus imaging window chamber is also integrated with the suction-based stabilized system for the stable in vivo uterus imaging. So thereby, all of these are fully optimized and provided as the final accessory product of IVIM technology. Through the IVIM intravital imaging platform, users can do the wide range of the preclinical research analyzing in vivo immune responses in the live mice model, tumor model, and the various human disease animal model. And also we can utilize intravital microscopy technology to monitor the drug delivery to the target cell in the live animal model, as well as validate the drug distribution in the target liver organ, and also in vivo drug efficacy validation in the skin and the tumor. And finally, IVIM's intravital microscopy has succeeded in high-quality live cell imaging in almost all of internal organ in the live mouse model. It includes the brain tumor, skin, pancreas, spleen, GI tracts, prostate, and some lymph node, memory tissue, placenta, and even heart and retina imaging in vivo. So from this slide, I'd like to introduce the intravital research application in the immunological research field in detail according to the target organ. This is the intravital imaging of your skin, visualizing immune cell dynamic in the skin of lysium GFP transgenic mouse, together with the ear skin blood vessels. As we can see here, we could visualize the cell-to-cell dynamic of the neutrophils in the skin, and also can acquire the very fast cell dynamic of the flowing neutrophils through the blood flow. This is another research example utilizing in vivo ear skin imaging technology. Here's the research example of the drug efficacy validation in vivo. So we successfully validate in vivo drug efficacy of the CARS protein, which is one of the secreted amino acid tRNA synthetase. To investigate how this enzyme induces inflammatory responses in vivo, we monitor the immune cell dynamic after intradermal injection of this red CARS protein to the ear skin tissue. Then the time-lapse in vivo imaging showed the active recruitment of the macrophage and the monocytes to the injected KRS, which finally enables in vivo validation of the inflammatory effects of the therapeutic material. For immunological research, we can directly monitor the immune cell dynamics in the palmatial lymph node. We actively transfer the T and B cells and monitor the extravasation dynamic of the T and B cells in high endothelial panel of the lymph node in real time. Here, the T cells and the B cells are selectively extravasated through the specific spots of HEB. And to monitor the stepwise transmigration process of the T and B cells in high endothelial panel, we perform the intravital 4D imaging of the T cell migration across the HEB. So it clearly showed the multiple steps of the T cell migration across the high endothelial panel, which are composed of the endothelial cell adhesion, intraluminal crawling, trans-EC migration, intraperivascular channel crawling, and the trans-FRC migration, and finally getting out of the HEB, arriving at the lymph node parenchyma. And from the long-term time-lapse imaging, we could identify that there are some hotspots in the endothelial panel where the T cell transmigration selectively occurs. For abdominal organs, we utilized abdominal imaging window for the long-term in vivo monitoring of the pancreas, spleen, and kidney. We could visualize the real-time cell dynamic of the monocytes and the dendritic cell in the spleen, and can also visualize the pancreatic islet cell in the pancreas. The cell dynamic and the development can be monitored in a long-term basis by utilizing this kind of abdominal imaging window. Especially in the kidney, we can do the real-time monitoring of the in vivo cell behavior of the neutrophil cell in the kidney. So from the real-time movie data, we can quantitatively analyze the cell velocity of each individual immune cell migration dynamic in real time. And for intravital lung imaging, by utilizing the suction-based lung imaging window chamber, we've succeeded in acquiring this clear and high-quality in vivo imaging of the lung, visualizing the neutrophils, vessel, and the blood flow dynamics simultaneously in the real time. So from this setting, we can perform in vivo microcirculation imaging of the neutrophil dynamic and real-time RBC flows in the live lung tissue. Also, we can apply this principle to the heart tissue imaging. This is the vacuum-based in vivo stabilized heart imaging chamber. With this system application, we can acquire this kind of real-time video of the heart, visualizing RBC flow in their blood circulation in the heart. Finally, we can monitor and track the flow of the individual single red blood cell and also leukocytes in the heart tissue in the live mouse model. And finally, for retina imaging, we customize the retina imaging system modified from the free-space system. So as the mouse eye has its own crystallized lens inside, additional optimized optic system is required for the retina imaging as shown in this picture. By utilizing customized retina imaging system, we successfully visualized various cells, including macrophage, retinal ganglion cell, microglia cell in vivo in the mouse retina. Sorry for the delays because the movie has the very big size. So yeah, now it's playing. So this is the real-time movie of the microglia cell dynamic in the mouse retina. We can analyze the movement activity of the microglia cell in the real time and can measure the dendritic movement velocity in the normal and also drug-treated mouse retina as well. The following is intravital microscopy application in the neuroscience research. For long-term in vivo monitoring of the mouse brain, we utilize the cranial imaging window to visualize the cerebral cortex. This is the research example monitoring brain neuroinflammatory responses induced by the 3-NP chemical injection. After induction, lysin-positive leukocytes are getting densely located to the specific size of the cerebral cortex within 10 days after inflammatory induction. Also, from the time-lapse imaging, we can visualize and analyze the meandering and crawling behavior of the neutrophils in the brain of the neuroinflammation mouse model. Then we can longitudinally observe the BBB integrity throughout 30 days of the neuroinflammation. So the vascular leakages and the following severe inflammatory responses with massive leukocytes infiltration at day 30 are reflecting acute BBB impairment in vivo. Also, we can visualize the neurovascular unit, which is composed of the astrocyte, parasite, and vascular endothelial cell in the cerebral cortex. So finally, we can analyze the BBB integrity, especially of the brain microvasculature integrity, by in vivo imaging analysis of the astrocyte, parasite coverage, and the vessel inside of the cerebral cortex. This is an exemplary research of in vivo monitoring of the neurovascular unit in the cerebral cortical area for 1 month. We can simultaneously visualize astrocyte and parasite composing the neurovascular unit. So in vivo parasite and vessel coverage with the cell dynamic of the division and the elongation process of the parasites were clearly monitored together with the identifiable astrocyte structures. And in cerebral microinfarction model, microinfarction in a penetrating arteriole were induced by the photothrombosis. So at 24 hours after the induction, a greatly increased level of the ROS and the cell death nearby the photothrombosis induction site were identified in vivo. To observe the cell level dynamic in the recovery of the parasite coverage following the microinfarction, a time-lapse intravital imaging of the NG2 positive parasite was performed for 24 hours starting from the 3 days after microinfarction. So the proliferation of the NG2 positive parasite and the process elongation along the vessels were clearly imaged, suggesting that the both processes contributed to the recovery of the parasite coverage after the microinfarction. To briefly summarize the research example in the brain disease model, including Alzheimer's, Parkinson's, and also neuroinflammation model and the stroke models can be tested in vivo for validation of the various type of the drug delivery, clearance, and the drug efficacy validation by visualizing the blood changes, immune cell dynamics, drug accumulation, and BBB integrity changes, etc. And finally, IVIM technology has wide range experimental in vivo research applications throughout almost all of organs in the live mouse model without any restriction of the imaging targets. And we also have a lot of research intravital research publication by using our IVIM system, which are categorized by the imaging target organ and the imaging purpose, such as blood cell flow analysis and in vivo validation of the drug and the cell delivery. Thereby, we can fully support the researchers with the optimized in vivo protocols and also with the strong academic backgrounds. For now, we're gonna quickly jump into the virtual demo. So for today's demo, we will perform in vivo liver and the brain imaging with the basic system operation. So we will gonna use both of the confocal and two-photon microscopy mode of the IVIM CMS3 intravital system. And for the software, there are two types of software we have. The IVIM engine is a imaging acquiring software, which is fully optimized for the real-time in vivo data acquisition. And the IVIM Studio is a post-processing software for the 2D and 4D image analysis visualization. So the Alex and Luna will show you the basic system operation using those two systems by acquiring the mosaic tiling and the 3D zigzag imaging. And lastly, the animal model for the liver imaging will be Lysan GFP transgenic mouse model, which is expressing GFP in the neutrophils. And moreover, we will additionally label the blood in the blue color by IV injection of the anti-CD31 PCAM antibody conjugated with the Alexaflor 647. And for brain imaging, cranial imaging window implant D6301 GFP transgenic mouse model has been prepared for the macrophage and the blood vessel imaging in the brain in vivo. So our application team members, Alex and Luna, have just prepared the experiment for the virtual demo session.
Thank you, Selina, for such an insightful presentation. Now we will move on to the live demonstration session. And I want to kindly remind everyone that the following content contains visuals of animals, real animals used in the research. I am pleased to introduce our application specialist, Alex, who will be conducting the live demonstration. And he will showcase the real-time intravital microscopic technique, as well as demonstrate the in vivo cellular level imaging in liver and brain. Alex, please proceed with the live demonstration.
Hello. Okay. Hello, this is Alex Kim, an application specialist from IVIM Technology. Today, I'm gonna show overview intravital liver and brain imaging by utilizing IVIM CMS3 model with Luna. Thank you, Luna. At the beginning, I'd like to introduce convertible confocal and two-photon microscope IVIM CMS3 model. Users can easily recognize the main body of IVIM CMS3, which has such a compact design compared to other devices. Beneath it, there are laser sources for confocal and multiphoton laser, of which wavelength is fixed to 920 nanometer. In this imaging room, terrace for 5 objective lens is furnished on the ceiling. Underneath the stage, we could see the 5 connectors connected to the rectal probe and plate heater, and so on. This is animal maintenance system with feedback functions from rectal probe and heating plate for stable body temperature of the anesthetized mouse. As for the surgery procedure of the liver, I will briefly explain about it. After an incision of skin and peritoneal membrane, liver of live mouse was mounted on the saline-soaked Kleenex, a disposable tower. And to start the imaging, I will slide the animal stage inside the imaging room. Let us jump into the imaging session with IVIM CMS microscope. Screen of IVIM CMS3 microscope will be shared for you in a second. Thank you, Luna, for helping us to film on-site demo with camera. So what you are seeing now in the shared screen is IVM Engine, an imaging acquired software for IVM CMS3. When you're closely looking at the panel on the left side, user interface seems to be quite user-friendly and intuitive, and actually it is. On the first row of the main screen, there are color-separated display for DAPI, RFP, and Sci-Fi channel, respectively. Underneath them, color-integrated live video and main display and window for the captured images would lie along the second row. So as to turn the microscope on, I will make the scanner on. Next, adjustment has already done for its laser power and detector values for an appropriate excitation and detection for in vivo imaging. Lastly, when I turn the shutter on and find the focal plane until the signals are on the display. It is totally feasible to see such a beautiful morphology of blood vessel on the digestive organ. Users can maneuver the jog dial, which get us to search for and to navigate the best field of view on this target organ. Those large branches of the portal veins with clear sinusoidal structure, along with the small arteries, may seem to be a good spot to mark and to take the signals from. When it comes to the fluorescence labeling, endogenous signal of the enhanced green fluorescent protein for leukocyte would be detected on the liver, and anti-CD31 antibody, which is fluorescently labeled with Alexaflora 647, was intravenously injected an hour ago in order that we could observe endocellular cell lining with pseudo-red color. And tissue infiltrated and small amount of circulating neutrophil with green color. IVIM engine makes it possible to image a number of combinations, such as mosaic for wide area imaging, G-stack for depth-away imaging, multi-position for multitude of spots, and time-lapse for temporal interval, as well as live video. As for this imaging session, I'd like to take the images with the combination of G-stack and mosaic mode from the liver. In order to do wide area imaging with IVIM CMS3, X and Y coordinates of field of view had to be set. This would be the first one, and that would be the last spot to be set. The number of images would be taken we will take for the entire area of interest would be 2 by 2 in a way of width and length. Additionally, on the same field of view, coordinates of G would be loaded. From the shallowest focal plane toward the deepest one, so that we could acquire depth-away imaging. Optical sectioning will be performed with the interval of 5 micrometer from top to the bottom surface, and the total imaging depth will be roughly 25 micrometer. During the image acquiring process of G-stack and mosaic, I'd like to share those imaging data that has been already taken in this morning. Every experimental conditions must be same. On the left side of the screen, 3 by 3 mosaic images in the width and the length way of liver. On the right side, there comes the extracted video of 3D build data. Look how beautiful in vivo liver imaging we could get from live mouse. I hope you could understand those limitations of us not being able to show every function in this short demo. IVIM Studio will be on the shared screen to post-process those acquired data from IVIM engine. This is 2D visualization mode IVIM Studio. for not only 2D post-processing, but also 3D, and furthermore, for the in vivo data analysis with the user-friendly UI. As I'm adding the high-state directory further with IVIM Studio, we could see the folder categorized files in abbreviated forms, and I will open it. From now on, I will change the adjustment of each color balance and brightness and contrast with the one of the single taken images of mosaic. Nextly, a set of JSTAG images would be played with the JSTAG and scale bar inserted. It is completely possible to perform basic functions, such as making G projection, and furthermore, quite a time-consuming and demanding one like mosaic, also called stitching. What you've already saw is just merely a part of what you can do with IVIM Studio. Don't forget there exists a number of possibilities of researchers utilizing IVIM microscopes, dependent upon their detailed scheme for in vivo experiment. My colleague Luna have already had the cranial imaging window mouse prepared and mounted on the imaging stage. Thank you, Luna. With the cranial imaging window implanted mouse mounted on the stereotactic plate with heating functions, we could observe the brain from meninges to the cortex with the imaging depths of 100 to 150 micrometer in case of confocal, and 200 to 300 micrometer case of two-photon microscope. As for the surgery procedure of cranial imaging window implantation, I will shortly put it this way. After an incision of skin and getting rid of some of the connective tissue just right on top of the skull and doing a bit of drilling, the circular cranial bone fragment of which diameter is 3 millimeter would be replaced with round cover glass which has the same diameter. And the other exposed skull would be molded with the pink dental cement. And to start the imaging, I will slide the animal stage inside imaging room. Let us jump into the imaging session with the IVIM CMS3 microscope. Screen of IVIM CMS3 microscope will be shared for you in a moment. Regarding the fluorescence labeling of cranial imaging window implanted mouse model, we can observe the tissue-resident macrophages and circulating monocytes with endogenous green fluorescent in the same green color. All the blood vessel lumen inside the brain would be filled with 40 microgram of advanced blue in red color. For now, I'd like to find the focal plane for the cranial imaging window, actually. As I already told you that 3 millimeter diameter cover glass is quite small area for us to find the focal plane of the brain, the cortex. So it takes probably, just give me 30 seconds. Okay, now you can see what you've got. As for this imaging session, I'd like to take the JSTAG images on the same field of view. coordinates of G would be safe. From the most upper focal plane toward the deepest one. Optical sectioning will be performed with the interval of 15 micrometer from top to the bottom surface, and the total imaging depth would be 100 micrometer from top to the bottom. During the image acquiring process of GSTAC, I'd like to share those imaging data already taken in this way. With regard to the experimental condition, endothelial cells are labeled with anti-CD1 antibody conjugated with alexaflora on red color. On the left side of the screen, 5 by 6 mosaic images in the width and the length way of brain. On the right side, there comes the extracted video of 3D data, which is the 21st mosaic image from the large map. You can see on the middle right side of the mosaic part, you can see clearly have the U shape, I mean S shape with the large branches structures in the cortex, you can see. Look how wonderful in vivo brain image we could get from the live mouse. Finally, it is time to post-process those acquired GSTAC data from the 3D visualization mode IVIM engine. Again, this is the 3D visualization mode IVIM Studio. Powerful tools for 3D and 4D in vivo data analysis. In this final chapter of our long journey, depth weight taken sequences would be utilized for 3D reconstruction. After a few adjustments of this one, I will make the video clip with the horizontally rotating 3D video for the complementary data, just the same as I show you during the imaging. So far, we have thoroughly looked into in vivo liver and brain imaging, of course with fabulous hardware and software. If there remain further questions regarding the experimental details of hepatic and cranial imaging and technical hardware area, please feel free to ask us in the next question session, or you could send us an email later on. Thank you.